Hi, I'm Jeff Mugridge. I'm an assistant professor here at UD. And in this video, I really just wanted to give a short, big picture overview of what my lab does. So my lab really studies how cells leverage chemical complexity on RNA to control gene expression. So the cell decorates its RNA molecules with this huge diversity of different types of chemical modifications. These range from relatively simple modifications like methylation or acetylation to much more complex modifications that add multiple chemical groups or larger functionality to these different RNA nucleobases. And so it turns out these modifications are really important and they can affect mRNA and tRNA structure, processing, translation, and RNA stability. And the RNA modifying enzymes that read, write, and in some cases erase these different chemical modifications are linked to really serious human diseases like different cancers and neurodegenerative disorders. And so my lab is really interested in understanding the chemical, structural, and molecular mechanisms that control these RNA modifying enzymes. So we typically work with purified proteins, and we use techniques from biochemistry and biophysics, like enzymology, NMR, X-ray scattering, techniques from structural biology, namely macromolecular protein crystallography, techniques and tools from chemical biology, and also bioinorganic chemistry to understand how RNA modifying enzymes work, how they recognize their target substrates, how they carry out their, in some cases, really unique and interesting biochemical reactions, and how they're regulated by different protein-protein and protein-nucleic acid interactions. And then our goal is to really take all of this high-resolution mechanistic information that we learn in vitro in the test tube to understand how RNA modifying enzymes impact RNA function in the cell and how these pathways are linked to important human diseases.